Hello, welcome to GK Possible IS. Today we will discuss regarding coronavirus and what type of questions might come from coronavirus related issues. So first we will see what will be the probable prelims questions and from which areas those questions might come. One is virus and bacteria because this is a coronavirus. Another is from DNA and RNA. Then various testing procedures like rapid antigen testing, RT-PCR and other testing like ELISA, all these things. Then what is the concept of pandemic and epidemic, what is the difference between them? Then World Health Organization, antigen and antibody, what are the antigens, what are the antibodies, sanitizer and disinfectants, what are the various chemical compositions and formulas. Then Epidemic Diseases Act 1897. First, coming to the coronavirus or COVID-19 virus, it is basically a SARS-CoV. It is known as SARS-CoV-2, that is severe acute respiratory syndrome, coronavirus 2, because there was another virus, SARS virus earlier also, and many coronaviruses till now have been also found out. It was, and this current coronavirus, that is COVID-19 virus, was first identified in 2019 in Wuhan of China, and it has been declared as a pandemic by World Health Organization. Then coming to why it is called as coronavirus. Why it is called as coronavirus? The main reason because this virus have crown shape. This virus, the outer coat is like a crown shape, like this here you can see, and this crown is a this corona means crown. Then coming to the various tests, one test is the rapid antigen test, in which we can get the reports or the result within 30 minutes. It basically detects the presence or absence of an antigen. Basically the antigen denotes the presence of a virus in a person. What are the antigen and what are the antibodies, we will come to that. Before that, it is a test, basically rapid antigen test is from the nasal samples. This is tested, and it's the success rate of rapid antigen test is low compared to the RT-PCR test. And if it comes positive, means it is true positive. That means if it comes positive, then you have definitely the coronavirus. But when it comes negative, it is not sure whether there is coronavirus or not in your body. For that, you can go for the RT-PCR. Now coming to the concept of antigens and antibodies. Now what are these antigens? Basically these antigens are found in almost all the living organisms, and these are usually proteins, and that is present in all the means in our bodies as well as externally also, especially in the coronavirus also. When that antigen of the coronavirus comes to our body or the coronavirus enters our body, our antibodies present in our body will start fighting with these antigens, or that means antigens are molecules capable of stimulating an immunoresponse. This is known as immunoresponse, and these antibodies are basically Y-shaped proteins produced by the B cells of our immune system in response to exposure to antigens, and these are the proteins, and these are present within the body, whereas antigens are present within the body or externally. And whenever these are the antigens and these are the these antibodies bind with these antigens, and so that these antigens cannot be active. When our antibodies are not sufficient or not capable enough to fight with these antigens enter our body, and they cause harm to our body. Then coming to the concept of DNA and RNA. First coming to the DNA, basically DNA, we can call them as the set. These DNAs are known as the deoxyribonucleic acid, and they contain the genes. These are the DNA strands, and in each pair they contain the basic genes. The genes are the basic building blocks of every organism, and these are present especially in animals, plant, protist, archaea, bacteria, and sometimes in viruses also. In viruses we can find the both plus DNA plus RNA. Whereas the ribonucleic acid or RNA is physically different from DNA. DM contains two strands, whereas RNA contains only one strand, whereas DNA contains two strands. RNA is the carrier of genetic information in certain viruses. As I said in the previous slide, DNA and RNA. Some viruses have DNA and some other viruses have RNA. We should know about this DNA and RNA before going to the RT-PCR test. Now coming to the RT-PCR test, this RT-PCR test stands for the reverse transcription polymerase chain reaction. Then first we'll know what is the polymerase chain reaction. Polymerase chain reaction basically a method or process in molecular biology to make billions of copies of a specific DNA sample from a very little amount of that DNA sample. Why this is important? Because through this polymerase chain reaction, for example, let's say we are collecting the nasal swabs from a patient and we will be testing it whether the virus is present in it or not. When in the laboratory it will be examined, that the very small swab may contain very less amount of that DNA elements or RNA samples of this virus, so it will not be visible or it will not be detectable in the laboratory. So what is done, through the polymerase chain reaction, it is converted, it is amplified, means from a little amount of DNA sample, billions of copies of DNA sample is created, and from that we can see easily which type of DNA or RNA samples are present in that. Basically, polymer chain reaction is for DNA sampling, and it was invented in 1983 by Mullis, for which he got the Nobel Prize, and it was a very important invention, because of which now this diagnostic test has improved a lot. And basically this PCR test, polymerase chain reaction, is used in many of the things like DNA cloning, then gene cloning, then monitoring the hereditary diseases, amplification of ancient DNA, analysis of genetic fingerprints for DNA profiling, then detection of pathogens, all these things. Then first, see, in this reverse transcription polymerase chain reaction, first we knew about the polymerase chain reaction. What is polymerase chain reaction? Now we will come to the transcription, then we will come to the reverse. What is reverse transcription? First, the transcription. Transcription is the process in which a gene's DNA sequence is copied to make an RNA molecule. When we convert a DNA molecule to RNA, it is known as transcription. But here, what is coronavirus? Coronavirus has RNA, not DNA. So now what we will do, we will convert RNA into DNA. So it is known as reverse transcriptase. And after that, we will sample the DNA through the PCR technique. So overall, it is known as the RT-PCR, reverse transcription polymerase chain reaction. Sometimes it is also called as real-time process. Then antibody test. Another test is known as the antibody test. It is also known as the serological test. Serological test or the serological survey. It is done to know it is done among a vast population to know how many people have been affected by this particular virus. And antibody, it just only says how many people have developed the antibodies in their bodies. Antibodies, what are the antibodies? We already have studied in the previous slides. Antibodies are the immunoresponse of our body to outside antigen or to the viruses. So this antibody test will show you how many people have been exposed to the virus. So basically, sometime it may happen that the antibody might have developed, but the virus is not present in our body. So it will not give the exact position how many people have been affected rather it or infected, rather it will be give the result about the how many people have been exposed to the virus. And basically this is done through a blood test or serum test, and one of the main popular test is known as the ELISA test. What is ELISA test? ELISA test is known as enzyme linked immunosorbent assay, and this ELISA test is especially used in HIV testing. It is also used for Zika virus and rotavirus. Now it is being done for this antibody testing or the serological survey. In earlier years also ELISA test has come in exam in UPSC prelim. So they might ask you a question like ELISA test is related to which of the following testing procedures, or for testing of Zika virus, rotavirus, HIV testing, all these things. Then coming to the sanitizer. Sanitizer is also important topic for this UPSC prelim exam, because many times we from the starting also we are discussing about these hand sanitizers, all these things we are reading in the newspapers also. Basically they might ask you the question regarding which of the following alcohols is used in hand sanitizer. Basically three important alcohols. One is isopropyl alcohol, another is ethanol or ethyl alcohol, another is N-propanol. So basically these three alcohols to be remembered. Then these surfaces, when we want to disinfect the surfaces, we use the sodium hypochlorite. The sodium hypochlorite, the chemical composition is NaOCl. Then coming to the virus. A virus is that concept. Okay, what is the difference between a virus and bacteria? A virus is a small collection of genetic code, either DNA or RNA. That means virus contains either DNA or RNA surrounded by a protein coat. This protein coat is known as capsid. A virus cannot replicate. One important thing about virus is virus cannot replicate alone. It needs a host cell. To replicate. To make copies of them. In the process, what they do, they kill the host cell and they replicate themselves. So that's why the virus, especially the virus infections, are more dangerous than the bacteria infections. Even viruses infect all types of life forms, including even they can infect the bacteria also. Various viral diseases like AIDS, COVID-19, measles, smallpox already have been eliminated from the world. Then coming to the World Health Organization, basically this WHO, it is guiding all the nations in this global pandemic situation, and this WHO was set up in 1948, was established in 1948, and this 7th April 1948 is also known as World Health Day, and its headquarters is at Geneva, Switzerland, and it's a specialized agency of the United Nations, and one of the important governing bodies of the World Health Organization is the World Health Assembly or WHO. The important thing is India has been elected to the WHO this year, and it will be holding the chairmanship of the WHO for 1 year, from May 2020 till May 2021. Then coming to the concept of epidemic and pandemic. Epidemic is a localized concept, localized or regional, especially in one region or one continent, whereas pandemic is a larger epidemic. It's a larger epidemic and it covers several countries or spreads from one continent to another. For example, HIV, AIDS, COVID-19, they can be termed as pandemics, and diseases like Ebola, Zika virus, chikungunya, dengue, all these can be called as epidemic. Then coming to the Epidemic Diseases Act 1897, it is a very old act, nearly 123 years old, and it was basically enacted by the British government in 1897 when the bubonic plague was there, and to control this spread of that plague in Bombay in 1890s, this act was enacted. And one interesting fact with this, that Bal Gangadhar Tilak. At that time, he questioned in his Maratha and Shivaji newspapers regarding the British government's response to the plague, for which under this act he was imprisoned. And this is basically one-page act, only one-page act containing 4 sections, and it aims to provide for the better prevention of the spread of the dangerous epidemic diseases. And it gives the state governments and union territories special measures to take special measures and formulate regulations for containing the outbreak. And recently the central government amended the act in April 2020 to we can at that time we could see many violences against the healthcare personnel, especially doctors, nurses. So to stop that, the central government has amended the act with imprisonment for a term of 3 months to 5 years and fine of rupees 50,000 to rupees 2 lakh. In case of causing very serious injuries, imprisonment shall be for a term of 6 months to 7 years and with a fine of rupees 1 lakh to rupees 5 lakh. Okay, thank you all, and please do subscribe my channel and like my channel. I will be uploading many new videos, especially for the UPSC and the other state civil service exams and the SSC exam. Thank you.